5 rules to tackle antibiotic resistance. Current efforts to tackle antibiotic resistance are not nearly radical enough, a leading scientist says. Dr. Ben Raymond, of the University of Exeter, says relying too heavily on reducing antibiotic use and discovering new drugs could lead to disaster. In a new study, he proposes 5 rules for sustainable use. These include acting to protect new drugs before resistance becomes a problem, using more diverse antimicrobials to reduce long-term use of single drugs, and using data to design management plans for particular superbugs. The World Health Organization says antibiotic resistance is rising to dangerous levels worldwide, and warns of a post-antibiotic era in which common infections and minor injuries can once again kill. Dr. Raymond, of the Centre for Ecology and Conservation on the University of Exeter's Penryn campus in Cornwall, said, "People think the best way to tackle antibiotic resistance is to give out fewer antibiotics and find new drugs. Those are important steps, but this approach alone is not nearly radical enough. Even if we can keep finding new drugs, disaster will follow if we use them in the same way as we use current ones. No drug yet discovered is evolution-proof, and the typical practice of using single drugs at once, in unprotected monotherapies, is unsustainable. This business-as-usual approach can be disastrous, as exemplified by the history of resistance in gonorrhea and the emergence of untreatable infections. Resistance to new antibiotics can become widespread in 2 or 3 years, so new drugs must be partnered with more sustainable patterns of use. The study's 5 rules are: 1. Prevention. Resistance is easier to deal with before it becomes severe, Dr. Raymond said. Antibiotics can be protected by the way you use them, for example by avoiding heavy use of single drugs for extended periods of time, as using drugs in this manner creates more selection pressure, the conditions microbes need to evolve resistance. 2. Don't rely on fitness costs. Some plans depend on stopping use of a drug, in the hope that resistant bacteria suffer a fitness cost, dying out because they carry resistance genes that are no longer useful. This can work, but Dr. Raymond warns that resistance to a drug does not necessarily go away just because use of that drug stops. 3. Limit supply of mutations. One way to do this is to use combinations of antibiotics, as microbes rarely develop resistance to multiple antibiotics at once. Dr. Raymond also says it's madness from a resistance management perspective to build up a massive supply of resistance genes in the environment. Resistance in the environment can come from wastewater and use of antibiotics in animals. As an individual, you are very unlikely to have acquired an antibiotic resistance microbe from an animal, but it's highly likely that environmental contamination has helped some of the microbes in your body acquire resistance, he said. 4. Low doses don't work, short courses might. A much greater pool of mutations can give microbes resistance to low doses of antibiotics, so such doses might help resistance evolve. Short, intensive courses of antibiotics might help patients without giving microbes the opportunity to evolve. 5. Information is power. If you don't know what kind of resistance is around among patients or in your hospital, you could give people the wrong drug at the wrong time, said Dr. Raymond. The more data you have, the better you can design your resistance management programs. Resistance management programs should target specific microbes or groups of microbes, rather than resistance in general. Dr. Raymond warns that broader lessons of resistance management from other disciplines are not widely appreciated among microbiologists, while evolutionary biologists and clinicians need to talk to each other much more often. The study, partly funded by the Medical Research Council, calls for a new philosophy in which usage is tied to a long-term commitment to sustainability. Dr. Raymond added, some humility in the face of natural selection can ensure that human creativity keeps pace with evolutionary innovation. Early in vitro testing for adverse effects on embryos' medications have to be safe for mothers-to-be and for their unborn children. Before the authorities will approve a new drug, it must be tested in animal trials on pregnant rodents and, as a rule, pregnant rabbits. Scientists in the Department of Biosystems Science and Engineering at ETH Zurich in Basel have now developed a test that allows them to examine a drug's embryotoxicity in cell cultures instead of animals. The new test does not yet replace the animal trials that are legally required as part of the medication approval process. However, as the new procedure is simple, fast, and inexpensive, researchers will be able to use it in the future to test a large number of drug candidates at an early stage of the development process. Substances that are harmful to embryos will thus be detected early in the drug development process and not only in embryo toxicity studies in animal trials. Because they are so expensive, animal tests are not conducted until much later in the drug development process with only carefully preselected potential drug candidates. As the new test weeds out unsuccessful drug candidates earlier, it may help cutting costs and reducing the number of animal experiments. Cell culture test with stem cells. The new procedure is an advanced form of the embryonic stem cell test, in which substances are tested in vitro on so-called embryoid bodies. These three-dimensional clumps of cells are formed from embryonic stem cells, in this case, from a mouse, and undergo the first stages of embryonic development over a period of 10 days. No viable embryos can be produced from these cells. Led by Professor Andreas Hierelmann, the research group has now expanded this embryonic stem cell test to include human liver tissue. There's a whole host of substances that are not toxic in their original form, but can be transformed into adverse substances by the human metabolism, especially by the liver, explains Julia Booz, a doctoral student in Hierelmann's group and lead author of the study, which has been published in the journal Advanced Science. In contrast to the conventional embryonic stem cell test, the newly developed test can detect substances of this nature. Body on a chip. From beginning to end, the new test takes place in its entirety on a single cell culture chip, which is equipped with various compartments. The compartments contain microtissue spheres, formed from human liver cells by the spin-off in spiro, and embryoid bodies, grown from mouse stem cells. The liver microtissues and embryoid bodies have a diameter of about 0.5 millimeter and are placed in different compartments, which are connected through microchannels to guarantee a constant liquid exchange between the different groups of cells. We're the first to directly combine liver and embryonic cells in a body on a chip approach, Booz says. Just as a pregnant woman's circulatory system links the metabolic processes in her liver with those in her developing embryo, this connected system ensures constant interaction between the liver cells and embryonic cells. Metabolites created by the liver cells, including metabolites that are stable for just a few minutes, can thus act directly on the embryonic cells, Booz says. She explains that combining the two in a single chip presents an improvement over other existing in vitro tests, which investigate the metabolization of substances in the liver separately from the effects that these metabolites have on embryonic cells. Booz goes on to describe another advantage of the new test. In contrast to tests on living pregnant mice, in our test, the substances are metabolized by human liver cells, in other words, just as they would be in the human body when the medication is administered. She points out that this is relevant, as humans and mice have different metabolisms. Further development for high-throughput testing. The researchers demonstrated the effectiveness of the new test with the help of cyclophosphamide. This chemotherapeutic drug has virtually no effect in its basic form, but the liver transforms it into a substance that is toxic to cells. 2 tests were run with cyclophosphamide, one included the newly developed liver embryoid body test, and the other involved embryoid bodies alone without liver microtissues. What these tests revealed was that a 4-fold lower concentration of cyclophosphamide was enough to have an adverse effect on the development of the embryoid bodies when liver tissue was present in the same environment. Now, the scientists need to refine the test further before it can be applied in drug development. They are paying particular attention to the materials that are used in the test in addition to how well the procedures can be automated. Automation would be necessary if the pharmaceutical industry or other researchers were to be able to deploy the test on a large scale for high-throughput screening of drug candidates. In addition, the scientists wish to develop a test that uses reprogrammed human stem cells, known as iPS cells, instead of mouse stem cells. Then they would have an in vitro test entirely based on human tissue. Alzheimer's therapy passes another important test. The Alzheimer drug candidate PRI-002 has successfully completed phase 1 of clinical research involving healthy volunteers. When administered daily over a period of 4 weeks, the active substance proved to be safe for use in humans. The next milestone will be the proof of efficacy in patients in clinical phase 2. In summer 2018, the drug candidate PRIO02 already proved its safety and tolerability profile in humans when administered as a single dose, up to the highest planned dosage. The drug concentrations in the blood reached the values that had previously been therapeutically effective in animal models. Now we were also able to demonstrate the safety of the compound after 4 weeks of daily administration, says Professor Dieter Wilbold, director of Jülich's Structural Biochemistry Institute (ICS6) and of the Institute of Physical Biology at Heinrich Heine University Düsseldorf. The study was conducted by Professor Michael Wolzt at the Department of Clinical Pharmacology of the Medical University of Vienna, in close coordination with Dr. Dagmar Jürgens of the ICS6. Neurocios GmbH, a contract research organization, carried out the monitoring of both studies. In recent years, many Alzheimer's drug candidates have failed in the crucial clinical phase 3, in which the efficacy and safety of the drug are confirmed. They did not achieve any improvement in memory performance and cognition in Alzheimer's patients. The amyloid beta protein, A-beta, was the focus of many of the failed drug candidates. There is certainly a good reason for this, as there is clear genetic evidence that A-beta plays a decisive role in the development of Alzheimer's dementia, says Dieter Wilbold. Either research attempted to reduce the formation of A-beta monomers from its precursor protein AP by inhibiting the cleaving enzymes involved, or monoclonal antibodies were used with the assumption that they would induce degradation of various forms of A-beta. The amyloid beta monomer is a naturally occurring protein in the body that is produced throughout life from the amyloid precursor protein, AP. These A-beta monomers are non-toxic and very rarely aggregate into an A-beta oligomer under normal conditions, as several A-beta monomers have to assemble in the same place at the same time, making this a very rare event. However, even rare events become more probable the longer the waiting period is. This is the reason why age is the most important risk factor for Alzheimer's disease. When the first A-beta oligomers have formed, they can multiply themselves and, unlike the monomers, they are highly toxic. They also impair the normal function of the nerve cells in the brain. In contrast to the much larger fibrils in the plaque deposits between the nerve cells, oligomers are highly mobile and cause damage anywhere in the brain. We considered a different approach for our drug candidate, because the principle that it is not a question of the formation of A-beta itself but of its aggregation into oligomers, has been known for several years, says Dr. Antje Willouwhite from the Institute of Neuroscience and Medicine, INM4, who has accompanied Dieter Wilbold's research for many years. The mechanism of action of PRIO02 follows a completely different approach than the previous drug candidates. It directly destroys the toxic oligomers without the involvement of the immune system, disassembling them into the harmless A-beta monomers. In addition, PRIO02 belongs to a completely new drug substance class, the so-called D-peptides. These consist of the exact mirror images of the L-amino acid residues normally found in proteins of organisms and are therefore not at all or very slowly degraded in the body. As a result, PRIO02 is stable enough to be administered orally, i.e. as a tablet or capsule. I have been watching the development of PRIO02 with great interest. Not only because it represents an interesting and new approach in the therapy of Alzheimer's disease, but also because it can be administered in tablet or capsule form, which is particularly advantageous for elderly people, says Professor Oliver Peters from the Charité in Berlin. The results of the preclinical efficacy tests with PRIO02 were also usually obtained with oral administration. We were able to show that mice with symptoms similar to Alzheimer's had an improvement in cognitive performance after treatment with PRIO02. The memory and cognition of the treated mice were significantly improved compared to the placebo group and could even no longer be distinguished from the memory performance of healthy mice, says Dr. Janine Cucci, scientist from Professor Wilbold's team. These recently published studies in very old mice with fully developed pathology and cognition deficits, carried out at ICS6, showed that the memory and cognition deficits were successfully improved even in these old animals. Clearly under non-preventive conditions, emphasizes Dieter Wilbold. The results from the successful tests of single and multiple ascending dosages of PRIO02 in healthy, voluntarily participating humans allow a further development of the drug candidate. Our next goal is the proof of efficacy in patients, says Dieter Wilbold. We hope to successfully take this path with the company Preavoid. Preavoid is a spin-off of scientists from Forschungszentrum Jülich and Heinrich Heine University Düsseldorf, which also has the know-how to develop other drug candidates for neurological diseases. The development of PRIO02 has so far been financially supported by the Helmholtz Validation Fund. The study that was completed now has received additional support through the Part the Cloud Translational Research Funding Award of the US-based Alzheimer's Association PTC 19605853. Preavoid is a very innovative company and I am happy to be a part of its further development, says Professor Detlev Riesner, a leading scientist in prion research who recently acquired shares of Preavoid. About Preavoid. Preavoid GmbH is a biopharmaceutical company founded in September 2017 as a spin-off of Forschungszentrum Jülich and Heinrich Heine University Düsseldorf. The company develops and markets innovative drug candidates based on D-peptides for the treatment of neurodegenerative disorders such as Alzheimer's, Parkinson's, ALS, Huntington's chorea, and various tauopathies. The most advanced drug candidate is PRIO02, which was developed to treat Alzheimer's and is about to be tested further in humans. The company's headquarters are in Jülich. Opioids, leading cause of pregnancy-related death in new Utah moms as the opioid epidemic rages on. One vulnerable group, new moms, have often been overlooked. A new study at the University of Utah Health found that drug-induced death is the most common cause of pregnancy-associated death in Utah. Postpartum women who have previously or currently struggle with substance abuse are at greater risk of overdosing. The research team explored the need for continued substance abuse counseling, access to naloxone prescriptions and mental health care in the year following childbirth. The results are published online in the May 9th issue of the journal Obstetrics and Gynecology. Death is the tip of the iceberg for moms who deal with addiction. These are the women that we can see, said Marcella Schmid, MD, MS, MA, an assistant professor of maternal and fetal medicine at U of U Health and first author on the paper. The point of this study is to determine when our moms are most vulnerable, and it's the postpartum period. In a retrospective study, 2005 to 2014, Schmid compared the characteristics of women who experienced pregnancy-associated deaths, death of a woman during pregnancy or within 1 year from the end of a pregnancy. She found that drug-induced deaths were the leading cause of pregnancy-associated death for women in Utah, with more than 3/4 of the deaths associated with opioids. 80% of deaths were in late postpartum period after mom has finished her last checkup with the obstetrician, Schmid said. We need to really look at fourth trimester care and how to enhance this care, especially women with substance use. According to Schmid, the vast majority of women will stop using or decrease using substances during pregnancy and are particularly vulnerable to relapse postpartum. Drug-induced mortality in the year following birth is often due to a combination of decreased tolerance following drug abstinence during pregnancy, hormonal changes, postpartum depression, stress and sleep deprivation from infant care, strain of intimate relationships and loss of insurance. The American College of Obstetricians and Gynecologists recently recommended that postpartum care for women be extended to a fourth trimester, typically longer than the 6-week postpartum check. In the study, the team identified 136 pregnancy-associated deaths using the Utah Perinatal Mortality Review Committee database. Of these cases, 26% were linked to drug use. Nearly 3/4 of the drug-induced deaths were associated with opioids, 27/35, 77%. Surprisingly, 42% of pregnant women insured through Medicaid in Utah are prescribed opioids for back pain, abdominal pain, headache or migraine, joint pain or other pain diagnosis. Drug-induced deaths, 35/136, 26%, were equal to the next two most common causes combined: blood clots, 18/136, 13%, and motor vehicle accidents, 17/136, 12%. The remaining deaths, 66/136, 49%, were associated with hypertension, infection, homicide, suicide, hemorrhage, malignancy and other causes. Although the majority of women in the study had drug misuse or substance use disorder in their medical files, none of the women were screened for drug use during prenatal, delivery or postpartum care. Fewer than half received a social work consultation, 14/35, 40%, or mental health counseling, 9/35, 26%. None of the women were taking buprenorphine or methadone to treat opioid use disorder. According to Schmid, this is not a Utah problem. Other states and cities that conduct maternal mortality review are finding similar increased drug-induced deaths in postpartum mothers. Women in the study who died from drug use primarily died at home, 20/35, 57%, compared to other pregnancy-associated deaths, who most commonly died at the hospital, 64/101, 63%. In addition, drug-induced deaths were more common during the late postpartum period, 28/35, 80%, defined as 43 days to 1 year after giving birth. Schmid notes the study is based in a state with a small population and does not include insurance status at the time of death or insurance lapses. It also lacks complete data on abuse or intimate partner violence and complete information about substance use treatment or social circumstances. We cannot do anything for the moms who died, but as a health care institution, we can identify the holes in care and plug them, Schmid said. We need to develop an addiction-knowledgeable obstetrics workforce. Schmid was joined on this project by Aaron Clark, Michelle Debink, Brett Einerson and Michael Varner of U Health and Intermountain Healthcare, Nicole Stone and Lori Bash of Utah Department of Health, and Adam Gordon of U Health and the Salt Lake City, Virginia Healthcare System. Dr. Schmid is supported by the National Institute of Health Women's Reproductive Health Research Career Development Program.